Now, first of all, I do want to welcome everybody to our webinar today, Dietary Fluoride Intake, Metabolism and Health. My name is Georgiana Alina, and I'm one of the Journal Relations Specialists of Nutrients. I'm here today to be the host and to present you a little bit more information about the webinar. Now, regarding MDPI, well, we are a pioneer in scholarly open access publishing. MDPI has supported academic communities since 1996. It is based in Basel, Switzerland, and MDPI has the mission to foster open scientific exchange in all forms across all disciplines. Now, our 477 diverse and open access journals, including 468 peer-reviewed journals and 9 conferences journals, are supported by more than 295,000 academic experts who share our mission, values, and commitment to providing high-quality service for authors. And we serve scholars from around the world to ensure that the latest research is freely available and all content is distributed under a Creative Commons Attribution license. Now, about the journal. Well, Nutrients is an international peer-reviewed open access journal on human nutrition, published semi-monthly online by MDPI, the Asia Pacific Nutrigenomics Nutrigenetics Organization, Italian Society for Pediatric Gastroenterology, Hepatology and Nutrition, Nutrition Society of New Zealand, and the Ocular Wellness and Nutrition Society, and others are affiliated with Nutrients, and their members receive a discount on article processing charges. Now we have high visibility by being indexed within Scopus, PubMed, Web of Science, Medline, PubMed Central, Agris, and other databases. The journal is ranked in JCR and SciScore in the Q1 in the category of nutrition and dietetics. Now, this is the program for today's webinar. We will first have our chair introduction. Dr. Ashley Malin will introduce us with the webinar details. We will have 3 speakers, Dr. Ashley Malin, Dr. Kyla Taylor, and Dr. Durdana Khan. And in the end, we will have the Q&A section where you as the participants could ask our speakers from today any questions, and in the end will be the closing of the webinar made by Dr. Ashley Malin. And this is a relevant special issue for today's webinar, Dietary Fluoride Intake, Metabolism and Health. If you are interested, you can check the special issue, and we are looking forward to receive some submission or for you to check the special issue. Now, regarding the Q&A section, you can add any question in the Q&A box that you can find it on the screen and just add the question. You can also mention if you have a particular question for each speaker, which speaker is the question, or just had a general question for the webinar. Now, I would like to introduce our chair from today, Dr. Ashley Malin. She is from the College of Public Health and Health Professions and the College of Medicine from the University of Florida, and she will be our chairs from today, and I would like to pass this to her. Thank you.
Hi, everyone. Thank you all for being here today. We're really excited to be hosting and speaking in this webinar, and it's great to see a nice turnout despite the short notice. So first, I'd like to introduce our guest speakers for today. I'm actually just going to share my slides. Okay, so today's webinar, as Alina mentioned, is on dietary fluoride metabolism and health, and this is the theme of the special issue in the journal Nutrients that I am a guest editor for. Okay, well, so I'm excited to be joined by Dr. Kyla Taylor, who is an epidemiologist at the National Institute of Environmental Health Sciences. She's also the lead author of the recently published meta-analysis on fluoride exposure in children's IQ that was published in JAMA Pediatrics earlier this year. I'm also excited to be joined by Dr. Durdana Khan, who is a postdoctoral fellow in my lab at the University of Florida. She's conducted research on fluoride exposure in early development and child neurobehavior and neurodevelopment in the US, and she's also lead author of a study we published earlier this year in Nutrients, which was the first publication on urinary fluoride levels among youth in the United States. And so Dr. Kyla Taylor, is going to be speaking about the National Toxicology Program's systematic review that was published last August, and also the recently published subsequent meta-analysis that, as I mentioned, was published earlier this year. And Dr. Durdana Khan is going to provide an overview of some of the work that she has been conducting on prenatal fluoride exposure and child neurodevelopment. And we are also, Dr. Taylor and I are going to be co-chairing a symposium at the International Society for Environmental Epidemiology and Exposure Science annual meeting in Atlanta in August, which is a week away. The symposium is on August 18th, and Durdana is going to be presenting on that symposium and also presenting a poster at that conference. She's going to give a sneak peek of some of that work today as well. All right. So for today's webinar, I'm going to give an overview of the special issue on fluoride metabolism and health, and talk about some of the focus of that issue, what the scope entails, and really what we're looking for in terms of submissions. Then Kyla is going to give an overview of the NTP monograph and subsequent meta-analysis and the research needs for fluoride and neurodevelopment. So she's going to talk about what the current state of the science on fluoride and neurodevelopment is, and then where there are still areas for exploration and, things that we don't yet fully understand. And then Durdana is going to give a brief overview of a recently conducted US-based work that she has recently conducted on fluoride exposure and infant neurodevelopment, as well as child autism-like traits. So both of those studies are conducted among women and children in Los Angeles, California. And then I am going to talk about research needs for fluoride and health outcomes aside from neurodevelopment. And then we have the question and answer. And I don't have any conflicts of interest or disclosures related to the material in this presentation. So first, I want to give a little bit of a background on fluoride exposure as a public health intervention. I'm not sure how familiar everyone is with fluoride, so just to give an overview, in Canada and the United States, fluoride is added to community drinking water for dental cavity prevention as a public health intervention. The recommended level for fluoride in drinking water is 0.7 milligrams per liter. So that's considered, as of now, the optimal concentration for reducing dental caries while also minimizing risk of adverse systemic health effects. Most of the United States does add fluoride to community drinking water for cavity prevention, although some states have stopped adding fluoride to drinking water, including Florida, which is where I live. And then different cities around the country have also stopped, in part due to concerns about potential neurodevelopmental risks that Kyla is going to talk about as well. In Mexico, fluoride is added to salt as well. In regions where drinking water fluoride levels are below that 0.7, and salt is also iodized, so iodine is added as well. Most of Europe does not fluoridate their drinking water, so some countries do add fluoride to salt or milk, but most of Europe does not add it to the community drinking water. And fluoride can also naturally occur in rock and soil, and then it can leach into drinking water from rock and soil. India and China have regions with high naturally occurring fluoride in drinking water. And these levels can be quite high. And when I say naturally, also sometimes fluoride can contaminate drinking water essentially through industrial processes inadvertently. Things like coal burning can cause fluoride to enter into the drinking water without necessarily adding it in intentionally. Okay, so for the special issue focus, we certainly welcome submissions that focus on dietary sources of fluoride, and I'll explain a little bit more what that means. But of course, given the title of the special issue, submissions on dietary fluoride, fluoride metabolism, also research that focuses on fluoride exposure assessment. So this is something that's important to think about, measuring fluoride in different ways across studies. Also, we welcome any well-conducted studies on potential impacts of low-level fluoride intake on systemic health outcomes. So there has been a lot of research historically on impacts of high fluoride levels, but in the last decade, there's been a lot more research on impacts of low-level fluoride exposure on brain development, but there's other health outcomes as well that we need research on potential impacts of low-level fluoride intake on. Also, any study that focuses on co-exposures of fluoride. So depending on the source of fluoride that an individual may be exposed to, a fluoride may also not occur in isolation. So whether it's fluoride from food or fluoride from drinking water, oftentimes there can be other co-exposures that occur along with it. Also, research that looks at interactions between fluoride and nutrient intake in relation to health outcomes is something that's within the scope of the journal and or the special issue rather, and quite suitable for the journal as well, given that the journal is called Nutrients. And I'm going to talk about what each of these aspects entail as well throughout this presentation, just to provide some more context. Okay. So in thinking about dietary sources of fluoride, there are many ways in which we can ingest fluoride through our diet. One of the main ways, particularly in a fluoridated community, is via foods and beverages that are made with tap water. So even though fluoride's added to the community drinking water, and turning on the tap and drinking a glass of water is a way to ingest that fluoride, all of the foods and beverages that are made with that tap water or processed with that tap water are then going to also contain fluoride. So it's even if someone, let's say, doesn't want to consume the tap water with fluoride and they have a filter, going to the grocery store and buying foods that are made with that tap water will then also provide a source of fluoride in the diet. As I mentioned, some countries do add fluoride to salt or milk, so that would be a dietary source of fluoride. Tea consumption is another one. Certain types of tea, such as green or black tea, have been shown to contain high levels of fluoride. I think more research needs to be done on that to see if that differs according to different brands of tea or different locations and things like that. But there is evidence showing that these types of tea can be high in fluoride. Certain pesticides can contain fluoride, and so if foods are sprayed with them, then they will also contain fluoride. Certain foods can also naturally be high in fluoride. Grapes, raisins, wine are examples. Seafood that retains bones. So fluoride has an affinity for our calcified tissue. And if seafood or aquatic life before they become seafood is exposed to fluoride in the environment, then that can be retained in bones. And then when we consume it, we ingest the fluoride from the bones. With that said, seafood, of course, has various health benefits. So it may be a matter of balancing those benefits with any risk from excess ingestion of fluoride. And there's also been research conducted in Mexico showing that pork rinds can be high in fluoride as well. So if anyone's doing work or thinking of doing work examining the fluoride content in different foods, that would be well within the scope of this special issue. Here's an example of a paper that I just mentioned, the one that looked at fluoride content in food in Mexico and found the high levels in the pork rinds. That was published, not in this journal. And then I actually co-authored this paper where we looked at dietary fluoride intake during pregnancy and found that it was associated with worse cognitive development in toddlerhood, particularly among boys. So these are just examples of studies that are on topics that could fit within the scope. And then this is a paper that was published in the special issue that examined associations between dietary patterns, fluoride intake, and excretion in women exposed to fluoridated salt. This was a pilot study looking at these associations and fit really nicely within the scope of this special issue. All right, I'm going to talk a little bit about fluoride metabolism as well right now. So fluoride, most of the fluoride that we ingest is absorbed in our bones, as I mentioned, and other calcium-containing organs and glands. So it has an affinity for calcium. The half-life of fluoride is relatively short, approximately 6 hours. Of course, that will differ depending on individual differences in physiology, diet, things like that. So roughly 3 to 10 hours for the half-life. And after we ingest fluoride, our plasma fluoride concentrations peak within about 20 minutes to an hour of ingestion. It gets absorbed into the stomach and the small intestines, and fluoride levels start to decline as it's absorbed in our calcified tissues and then excreted in our urine. So fluoride is renally cleared. We excrete it in urine via the kidneys. And the fluoride concentrations in urine will reflect the levels that are in our plasma, in our blood, as well as fluoride that's, in other organ systems or our calcified tissues like our bones that can be released in urine as well through bone remodeling, which is a lifelong process. Okay, so as I mentioned, another focus of the special issue is on fluoride exposure assessment. Urinary fluoride is the most widely established biomarker in studies of fluoride in health. So it's widely used in a lot of really well-conducted studies. Other studies also look at water fluoride concentrations, which can help to provide a sense of how much fluoride in the drinking water specifically may be contributing to a health outcome. Some studies have looked at plasma fluoride levels. I've published several studies that include plasma fluoride biomarkers, particularly among children and adolescents in the National Health and Nutrition Examination Survey. So they publish plasma fluoride levels. And some studies include community fluoridation status, which looks at do health outcomes differ according to whether someone lives in a fluoridated versus non-fluoridated community. Not as precise a measure as looking at a biomarker or specifically household water fluoride levels in terms of assessing fluoride exposure at the individual level. And fluoride, as I showed in those previous studies, can also be measured in food, or it can be measured in medication. Some pharmaceuticals also contain fluoride. Fluoxetine is an example of that, which is an SSRI, an antidepressant medication. So some of the benefits of looking at urinary and plasma fluoride biomarkers are that they capture fluoride intake from all sources, and they are moderately associated with tap water fluoride levels, and that we know tap water fluoride levels are the main source of fluoride exposure in the United States for adults especially, and also the contribution to total fluoride intake increases when we think about all of the foods and beverages that are also made with the fluoridated tap water. And plasma fluoride is also a beneficial biomarker in the sense that it may approximate the fluoride that's actually being retained in the body. prior to it being deposited in calcified tissue, whereas urinary fluoride may better reflect fluoride excretion. However, these biomarkers also have some limitations. So a challenge with fluoride having a relatively short half-life is that for plasma fluoride, it can be difficult to capture that snapshot of exposure. There is the potential for exposure misclassification in terms of there being measurement error or imprecision in the biomarker. And this has the potential to essentially create noise in the measurement, as I said, imprecision, which would bias associations toward the null. So it poses a limitation in terms of capturing an association because it may be weaker. What we capture may show a weaker association than what there actually is. Oops. And another limitation of these biomarkers is that they can be influenced by daily behaviors, meaning food and beverage consumption patterns. So if someone drinks a large cup of black tea right before getting a sample collected, then that can create a potential spike in fluoride intake. With that said, if that person regularly consumes black tea, then that spike may be a common one they encounter and reflective of their typical fluoride consumption patterns. But the influence of daily behaviors on biomarkers for fluoride exposure assessment can be mitigated by collecting a fasting sample. So have someone provide a urine sample before they eat or drink anything, or collecting 24-hour samples, for throughout the day, basically. Fluoride exposure biomarkers can also be influenced by differences in metabolism. And so that's something important to keep in mind when researching health effects of fluoride exposure and using different biomarkers to address or adjust for factors that might contribute to differences in metabolism. And some other fluoride exposure biomarkers that are being used include assessing fluoride in toenails or fingernails, assessing fluoride in teeth. So looking at fluoride levels in tooth dentin, some of our colleagues are now looking at that. Also assessing dental fluorosis, the presence of dental fluorosis or severity of dental fluorosis, which is essentially changes in the development of the tooth structure and tooth enamel due to excess fluoride exposure during tooth development. And fluoride can also be measured in hair. And as I mentioned, me and Kyla are going to be co-chairing a symposium next month on August 18th at the joint meeting for ISCE and ISCS in Atlanta. And that will focus on new research on early life fluoride exposure and neurocognitive outcomes across the life course from US and Canadian studies. We have presentations. Jordana is going to be presenting on this symposium, as I mentioned, and presentations that include biomarkers ranging from urine fluoride to toenail fluoride to dental fluorosis, and examining those exposures in relation to neurocognitive outcomes. So if anyone is in Atlanta or around that area next month, we invite you to please come join our symposium. And with that said, I'm going to turn it over to Dr. Kyla Taylor to talk about some of the work that she has been conducting on fluoride exposure and neurodevelopment over the last several years.
All right, thank you so much. Let's see, let's get my slides. Okay, so yeah, thank you so much for that introduction. I'll be talking about our systematic review and meta-analysis on fluoride exposure in children's IQ. I'll start with a brief background on my division and my branch at the National Institute of Environmental Health Sciences. I'm in the Division of Translational Toxicology, and our mission is to improve public health through data and knowledge development in a way that's translatable, predictive, and timely. I'm in the Integrative Health Assessments branch. We conduct systematic reviews and hazard identification assessments. I'll be walking through our methods briefly during the talk today. Our work is used to inform public, government, scientific, and medical communities, and we rely on the highest standards in rigor and reproducibility. Our products include the congressionally mandated report on carcinogens, NTP monographs, systematic reviews, systematic evidence maps, etc. So all of our assessments rely on systematic review methodology, and this is a way to increase transparency, rigor, and decrease potential for bias. The OHAT approach is an approach we developed in 2014 as a framework for systematic review and evidence integration across human, animal, and mechanistic studies. These are state-of-the-art methods, and OHAT has led the field in bringing these standards to toxicology and public health. And given the importance of public health decisions, it's essential to follow standardized methods and be transparent in reporting. So Ashley covered what fluoride is. This is just a brief slide showing the different topical sources and systemic sources. It's a mineral that does occur naturally in some community water systems. Dental caries are a result of when sugars that are contained in food and drinks are converted by bacteria into acids that demineralize the enamel. And so the presence of proteins and ions like fluoride in the oral environment can neutralize these acids, and it helps to slow demineralization and promote remineralization of the enamel. Topical fluoride exposure, like brushing, delivers fluoride to the interface between the tooth surface, the enamel, and the oral fluids, and systemic exposure, such as fluoridated drinking water, can also have anti-caries effects through contact with the enamel when it enters the oral cavity and when it's absorbed in the recirculated saliva. I also wanted to mention total fluoride exposure in this talk refers to the fluoride ingested from all sources. So not just drinking water, that includes systemic sources or any topical sources that might be swallowed. And urinary fluoride measurements are a valid estimate of total fluoride exposure. And as Ashley mentioned, this can vary widely by individual based on their behaviors and habits. So as background on our work, in 2006, the National Research Council concluded that there was evidence of neurotoxic effects of fluoride in humans and animals. In 2015, the topic was nominated to the NTP for evaluation. And in 2016, NTP published its first monograph on the experimental animal studies and found low to moderate evidence of adverse effects on learning and memory. And last August in 2024, we published a second monograph that included the human, animal, and mechanistic studies. We screened over 14,000 studies and identified over 500 that were relevant to our research question. 2 independent reviewers critically assessed study quality based on criteria that was predetermined, and all of the data has been documented and is available online. Based on epidemiological evidence, we concluded with moderate confidence that there is an inverse association between fluoride exposure and children's IQ. So that was a qualitative assessment. Today, I will be talking about a quantitative assessment, which was published in 2025, January of this year. This was a systematic review and meta-analysis that was published in JAMA Pediatrics, and it's the most comprehensive and transparent analysis of the children's IQ studies to date. So the OHAT approach for systematic review is outlined on the left-hand side of the slide. So I'll briefly be walking through this as I tell you more about the methods of our meta-analysis and that we used. So first, we developed a protocol for the meta-analysis with input from technical experts. After a formal peer review, it was posted to the NTP website for transparency purposes. Next, we identified the evidence, which has 2 parts. The first is we conducted a comprehensive literature search of 8 different databases, which was updated through November 1st of 2023. Next, 2 independent reviewers screened references for relevance. Study selection is based on predefined inclusion and exclusion criteria to avoid bias. Another layer of transparency is our interactive reference flow diagram. This includes links to all of the studies that were excluded during screening, along with the rationale for the exclusion. Our database searched, over 50,000 references, 30,000 of these were unique, and after screening, we had identified 74 studies of fluoride and children's IQ. So next we evaluated the evidence, which also has two parts. First is data extraction. We extracted data from each study into an online open source application that's called HAWK, the Health Assessment Workspace Collaborative. And also for transparency purposes, all the data that we extract is publicly available so that other researchers can replicate and extend our work. So the next and final step in the systematic review process is the risk of bias assessment, which is an evaluation of a study's internal validity. We followed a peer-reviewed protocol, and two independent reviewers critically assess each study's risk of bias, which helps us identify studies that are higher and lower quality. The higher quality studies represent the best evidence. Our method includes 7 risk of bias domains for observational studies, and studies that are determined to be high quality had to meet predefined criteria across each of these domains. For each individual study, we provide a risk of bias rating to each of the domains, which you can see on the right side of the screen. They range from definitely high to definitely low. We do this assessment also in HAWK, so that it's very transparent how studies got these different ratings. The first 3 domains that are listed with the green check mark are what we consider to be key risk of bias domains. So these are the types of bias with the greatest potential to impact results of a study. So for confounding bias, as an example, high quality studies that were found to be low risk of bias for confounding had to account for key confounders like age, sex, socioeconomic status, and co-exposures to other neurotoxicants like lead and arsenic. The remaining domains are also very important for identifying any other concerns that could indicate serious risk of bias issues. So we conducted a targeted analysis of 74 studies, and 22 of these were found to be high quality. Studies reported group level and individual level data, and fluoride exposure was estimated primarily in water and urine. We conducted 3 different meta-analyses. The first 2 used group level data, and that compares children living in high fluoride exposure areas to children living in low fluoride exposure areas. And the third analysis included studies that had individual urinary level data. So I'll be showing you the results of each of these analyses. The first was the mean effects, which included all studies using group level data. And to show you this, I'll be presenting a forest plot of the standardized mean differences, which we calculated for each study. So right now we are looking at the 47 low quality studies, and on the left side of the figure are the citations, and on the right are the SMDs. You can see the solid black line is indicating where SMD equals 0. We call this the no effect line. If an SMD equals 0, it means that there's no difference in IQ, mean IQ, between the 2 exposure groups. So you can see that almost every study, with a few exceptions, has an SMD that falls to the left of that solid black no effect line. And that means we're looking at a consistent pattern of inverse associations. So the pooled SMD for the low quality studies means children in the high exposure group had a mean IQ that was roughly 7 points lower than children in the low exposure groups. When we look at the high quality studies, there are 12, you can see that children in the high exposure group have a mean IQ that was roughly 3 points lower than the low exposure group. And there's the continued pattern of inverse associations is still there with the high quality studies. So next we calculated pooled dose response curves using linear and nonlinear models. And I'll be showing the results of the linear models, which were the best fit for the data. This analysis was done separately for fluoride measured in urine and water, and that's an important distinction, as I noted before, because urine is a valid estimate of the total fluoride exposure. So it captures fluoride from all the sources, including drinking water. And let's see. So here we're looking at the results right now. The water and fluoride columns are on the right side of this table, so the results for those are shown separately. when we looked at both high and low quality studies, so all studies at all exposure levels, we found that a significant inverse dose response with an SMD that roughly translates to a decrease of 2.2 IQ points for both fluoride measured in water and urine. When we restricted to only the high quality studies, we saw similar inverse associations. And we also wanted to look at the dose response relationship at lower fluoride levels. So to do this, we progressively truncated the high exposure group to be less than 4, less than 2, and less than 1.5 milligrams per liter. And these correspond with current drinking water safety guidelines. When limited to the high quality studies that had exposures at less than 1.5, we still saw inverse associations in both water and urine. And these roughly translate to a loss of between 1 and 5 IQ points. And I'll note that the association with urinary fluoride remains statistically significant, and the water fluoride did not. So our third meta-analysis, we calculated pooled regression slopes, which are also called beta coefficients, from 13 studies with individual level urinary data. You can see at the top are the 2 low quality studies, which had a pooled estimate of negative 3.38 points per milligram per liter increase in urinary fluoride. And among the 11 high quality studies, for every 1 milligram per liter increase in urinary fluoride, there is a significant decrease of 1.14 IQ points in children. So 3 of the studies that I just showed you before have maternal fluoride measurements that capture prenatal exposure. And that's important because developing brains are especially vulnerable to neurotoxicants. Each of these studies took multiple urinary samples throughout pregnancy and assessed IQ around age 7. And these are highlighted here in the forest plot. The first 2 that are highlighted in yellow, the Canadian and Mexican cohort studies, have maternal urinary levels that are comparable to levels in the United States. So these are the most comparable to fluoride that's added to drinking water in the US. And in Denmark, there's no community water fluoridation. So fluoride exposure levels in that study are lower than what we see in the United States, unless people in the US are living in areas with no fluoride added to water and no naturally occurring fluoride, which there are in some places. So we pooled these 3 regression slopes, which resulted in a beta coefficient of negative 1.7, which was not statistically significant. A recent study by Grandjean et al., they actually were able to pool all of the individual level urinary data from each of the 3 cohorts, and they found a similar effect estimate of negative 2 IQ points, which was statistically significant. We did conduct sensitivity analyses of other studies that measured maternal fluoride exposure, but they had outcomes that were not directly measuring IQ, so they used other cognitive tests. And when we included those studies in our analysis, we found that the effect estimates, it did not impact the overall results, and they remained inverse and significant. So overall, we took multiple different approaches for assessing the data and consistently found statistically significant inverse associations between fluoride exposure and children's IQ. These associations held when we restricted to only the best available evidence, which was the high quality studies. And we also found a linear dose-response relationship across all data and when restricted to studies with exposures at less than 1.5 milligrams per liter in both urine and water. And that's important because it means we're not relying on just one estimate of exposure. The biggest takeaway was the consistency and robustness of the inverse association across this large body of literature. It remained consistent across widely different study populations from different countries with different exposure levels and sources, both group and individual level data, and when fluoride was measured in both urine and drinking water. And so in systematic review and hazard assessment, a dose-response relationship and a high level of consistency in the direction of association strengthens our confidence in that overall association. Additional research would greatly help, would add to what we know right now. So as an example, high-quality prospective birth cohort studies that take multiple measures across pregnancy when they're estimating fluoride exposure that are conducted in areas that have lower levels of fluoride in drinking water, such as areas in the United States, would be very informative, especially to dose response at low levels. Ashley talked a bit about the biomarkers. We can always use improved exposure assessment methods, and there are researchers right now that are looking into biomarkers that capture better long-term exposures, such as teeth, hair, and nails. We also need to better understand the potential mechanisms. There's some data on this, but it's not right now. It's not conclusive on what those mechanisms are with neurodevelopment. And also, this isn't research, but it is something that is needed, is improving the communication strategies, using effective communication strategies for advancing fluoride research among scientists and the public and mitigating potential health risks. And so I'd like to thank my NIEHS and ICF co-authors for their incredible work, and I appreciate your time and attention. Thank you.
Thank you, Kyla, for that excellent presentation. Now Jordana is going to share some of the research that she's been conducting to address some of the gaps that Kyla mentioned.
Thank you. Let me share my screen. Thank you, Dr. Mellon, for your kind introduction. Hello, everyone. Welcome you all. Truly grateful for this opportunity to be here today, sharing our work. The project that I'm presenting today are collaboration between University of Florida, Department of Public Health and Epidemiology, and Department of Population and Public Health Sciences at University of Southern California. I'm going to discuss two research projects today, exploring the association between prenatal fluoride and infants' neurodevelopment and behavioral issues among children. Before we dive into the project's methods and findings, I would like to discuss the context and rationale behind these research projects. Concerns about fluoride's impact extend beyond dental health, particularly regarding neurodevelopment. There is some emerging literature linking fluoride to neurodevelopment and neurobehavioral issues. In the next few slides, I will be discussing some of the key findings of these studies. This foundational study from Mexico demonstrated a significant inverse association between maternal fluoride exposure and cognitive performance in children. They followed children at two key developmental stages, age 4 and between ages 6 and 12. The study measured fluoride levels in maternal urine during pregnancy as a biomarker of exposure. And this is important because fluoride can cross placenta, potentially affecting the developing fetal brain. They assessed cognitive function using a standardized tool, the Journal Cognitive Index, GCI index, at age 4, and IQ test at later ages. The findings were consistent across these two age groups, reinforcing concerns about early life fluoride exposure. Building on their earlier work, the same authors found that higher fluoride exposure during pregnancy was also associated with more pronounced ADHD symptoms in school-age children. This suggests potential neurobehavioral effects beyond general cognition. These findings suggest that fluoride may act as a developmental neurotoxicant, particularly affecting behavioral regulation system in the brain. Another landmark study based in Canada observed similar associations. The study was conducted within the framework of maternal infant research on environmental chemicals, MyRIC cohort, a well-established longitudinal study cohort. Children's cognitive abilities were assessed at age 3 to 4 years. using a standard testing protocol, which included both verbal and performance components to capture a comprehensive measure of intellectual performance. The findings suggest that male offsprings may be more vulnerable to the neurodevelopment effects of fluoride exposure in utero. This study added international weight to the evidence base. While previous studies have linked prenatal fluoride exposure to cognitive deficit in older children, this investigation aimed to determine whether such effects are observable earlier in life, specifically during the first 2 years of postnatal development. Maternal fluoride exposure was estimated based on dietary intake assessments during pregnancy, providing a comprehensive measure of fluoride consumption from both water and food sources. The study found that male infants whose mother had higher fluoride intake during pregnancy exhibited poorer cognitive development during the first 2 years of life. Although this association was not observed in female infants, this suggests potential sex-specific vulnerability to fluoride exposure during critical periods of brain development. Given the increasing evidence linking fluoride exposure to neurodevelopmental outcomes, this Canada cohort-based study aimed to determine whether prenatal exposure from community water supply could influence executive functioning in early childhood. Executive functioning refer to a set of high-order cognitive processes, including working memory, inhibitory control, and cognitive flexibility, which are critical for goal-directed behavior and academic readiness. Executive functions critical for school readiness and self-regulation. These findings highlight the broader cognitive domains potentially affected by fluoride. Given the findings from studies in Mexico and Canada that I discussed, and the lack of research within the US population, there is a significant gap in the literature. Specifically, there is a need to explore the effects of prenatal fluoride exposure on earlier stages of neurodevelopment, as early as 6 to 18 months of age. And it is plausible that early life exposure like fluoride may affect the fundamental biology of neurodevelopment, and these effects need to be carefully studied and quantified. So our first project focuses on early motor development. Specifically, we aim to assess whether prenatal fluoride exposure is associated with motor neurodevelopmental outcomes in infants at 6, 9, 12, and 18 months of age. For this, we utilize Ages and Stages questionnaire, a validated developmental screening tool, which I will be discussing shortly. Now I would like to discuss 2 USA-based studies that looked into fluoride and neurobehavioral outcomes. A first and recent cohort study investigated the association between prenatal fluoride exposure and neurobehavioral outcomes in children living in optimally fluoridated areas in the United States. Researchers followed 229 mother-child pairs measuring fluoride exposure through maternal urine samples during pregnancy and assessing child neurobehavior, which was quantified using the Preschool Child Behavior Checklist, CBCL tool, which included composite scores for total problem, internalizing and externalizing problems, and one of the subscales also particularly measured autism spectrum problems and ADHD problems. Another recent longitudinal cohort study of Florida Medicaid data followed over 73,000 children and found that exposure to fluoridated community drinking water over the first 10 years of life was associated with risk of neurodevelopment and behavioral disorders. These findings suggest that even in communities with recommended fluoride levels, prenatal exposure may carry risk for child neurodevelopment, warranting further investigation. Emerging evidence from these 2 US-based studies that I just discussed has reported association between fluoride exposure and autism spectrum disorder, suggesting a potential link that merits further exploration. We believe that there is still lack of comprehensive studies examining the effects of fluoride exposure on neurobehavioral outcome. So our second project shift focus to later childhood, where we investigate the potential link between prenatal fluoride exposure and ASD-like traits in school-age children. This allows us to explore both early and later neurodevelopment outcomes in relation to the same environmental exposure. Next, I would like to discuss the methodology of our projects. Our both projects utilize data from maternal and developmental risks from environmental and social stressors, commonly known as MADRS cohort. So MADRS is a prospective longitudinal pregnancy cohort, meaning we followed mother and child and children over time. Our analysis include mother-child pairs for whom we have available data on both exposure and outcome. The MADRS cohort provides a rich and diverse data set, making it an ideal foundation for investigating environmental exposures such as fluoride and their potential health outcomes. The MADRS cohort focuses on predominantly Hispanic women and their children of low socioeconomic status in urban Los Angeles. Participants were included if they were at least 18 years of age, less than 30 weeks gestation, and fluent in English or Spanish. Women were excluded if they have any disability that can prevent them from participation, being HIV positive, incarcerated, or having a multiple gestation pregnancy. We use a standardized method to collect the exposure data, which include maternal urinary fluoride levels. We employed specific gravity-adjusted maternal urinary fluoride to account for variation in urine dilution, thereby enhancing the accuracy and reliability of the exposure assessment. Urine samples were collected at 2 time points, first and third trimester of pregnancy. Maternal urinary fluoride is considered the most widely employed and validated measure of fluoride exposure in epidemiological research. It reflects total fluoride intake from all sources, including drinking water, diet, and dental products, making it a comprehensive indicator of maternal exposure. The outcome for our first project is the Ages and Stages questionnaire, which is a validated, widely used tool for monitoring child development in children aged 3 to 61 months. One of the distinguishing features of ASQ is that it is completed by parents, leveraging their careful and continuous observation of their child's behavior and abilities. It has high test-retest reliability and strong inter-rated reliability between parents and professionals, ensuring consistent and accurate results. The 5 domains of neurodevelopmental milestone that is included in ASQ screening is gross motor, that involves use of large muscles, activity like running, crawling, walking. Fine motor focuses on coordination of small muscles in the hands and finger. Problem solving relates to how child interact with toys, solve problems or puzzles. Personal social focuses on child's developing relationship and ability to perform personal tasks independently, such as feeding themselves or using the toilet. Communication involves the acquisition of communication skills, including receptive language, listening, understanding, communicating ideas or thoughts. For our second project to evaluate ASD-like traits, we used Social Responsiveness Scale, second edition, or SRS2. It's a 65-item questionnaire completed by parents designed to capture a wide range of behaviors and social characteristics associated with autism spectrum disorder. The assessment was conducted when the children were 5 years of age, a critical developmental stage for identifying early social and behavioral patterns. 5 domains of SRS evaluate social functioning across 5 core domains, each of which reflects a distinct aspect of social responsiveness. These domains are designed to capture both strengths and challenges in social behavior, providing a comprehensive profile of an individual or child's social abilities. The 5 core domains of social functioning include social awareness, cognition, communication, motivation, as well as restricted and repetitive behavior. Together, these domains provide a comprehensive profile of a child's social responsiveness and potential ASD-like traits. In our analysis, we accounted for several potential confounding factors, which we categorize as maternal and infant factor. We consider several maternal characteristics that may influence both exposure level and child developmental outcomes like maternal age, race, household income, and BMI. We also included key infant characteristics to adjust for developmental variability like sex of the baby and age at the time of assessment. This slide will provide a visual overview of our study design of our both projects, starting with MATRIS cohort of pregnant women. The exclusions are applied at this stage. We measure maternal urinary fluoride at first and third trimester. Maternal related covariates are examined at this time point and recorded. We followed pregnant women till newborn are there. So we have mother and child pairs. Baby factors or covariates related to infants are measured at this time point. For our first project, we measured outcome, that is ASQ score at 6, 9, 12, and 18 months. Over here, we did two types of analysis. We also looked longitudinally, where we incorporate all time points. And also we look time specifically individual at each time points, like 6, 9, 12, and 18 months of age individually. For our second project, our outcome is SRS scores that is measured at 5 years of age. Our statistical approach integrates both descriptive statistics and graphical techniques to ensure comprehensive understanding of the data structure and potential relationships. Our statistical analysis began with descriptive analysis to understand characteristics of our study population and distribution. This include calculating frequencies, mean, standard deviation, medians, and interquartile ranges. Also, we visually assess the distribution and relationships among variables. We employed several graphical techniques, which include histograms, joint scatter plots, and other Loess smoothing graphs. This box plot provides a clear visual summary of the distribution of maternal urinary fluoride values during third trimester. So the median maternal urinary fluoride concentration was 0.79 milligram per liter, indicating moderate variability in exposure levels among participants. This visualization of first and third trimester maternal urinary fluoride levels help us understand the distribution and frequency of exposure level in our study population. You can see the clustering of data at low fluoride values suggest that majority of participants had minimal exposure during the first and third trimester. The steep decline in frequency at higher values indicates that elevated exposure is uncommon in our cohort. Similarly, this scatter plot also shows that majority of the data points cluster around lower values on both axes. This suggests that most participants had relatively low level throughout pregnancy. There is some spread toward higher levels, but fewer cases show significant trimester-specific differences. This histogram shows the distribution of our ASQ score, that's the outcome for our first project. The most prominent feature is the peak at score 0, indicating larger number of observations at 0 score, indicating better performance. So lower score show better performance. At the score increase, observation declines sharply. This suggests a skewed distribution heavily weighted toward lower scores. These are the distributions of SRS score. That's the outcome for our second project. And the slide shows that for across all 5 domains, there's a skewed distribution heavily weighted toward lower scores. Lower scores mean better performance. To summarize our multivariate analysis, for our first project, we conducted both longitudinal analysis and a secondary analysis. It's a time-specific analysis. For both analysis, We use negative binomial regression models. We reported incidence rate ratios with 95% confidence intervals. And for our second project, we conducted linear regression models, and we report beta coefficients with 95% confidence intervals. We have interesting findings, and we are excited to share these results and interesting findings, and we are planning to present them in upcoming ISES joint annual meeting in Atlanta. We invite you all to please come and join us where we share our results and findings. Thank you so much. We are happy to answer any question in the end.
Thank you, Jordana, for that excellent presentation. All right, I'm just going to share my slides now. Kyla and Jordana talked a lot about, impacts of fluoride exposure on the developing brain, but there are other health outcomes related to neurodevelopment and also fluoride metabolism that I want to highlight today, where I think research is needed, particularly for low-level exposures. So one of those is the thyroid gland, and this is closely linked with neurodevelopment, particularly during the prenatal period when the developing baby is dependent on the mother's thyroid hormone health for brain development. And sorry, I don't know if anyone else can hear that, some interference, but if someone could just mute themselves, who isn't muted already, that'd be great. Thank you. So fluoride has been identified as an endocrine disruptor. There was a 2006 National Research Council report that concluded that fluoride is an endocrine disruptor in the broad sense of altering normal endocrine function or response. And research shows that fluoride can interfere with enzymes and hormones that are important for thyroid function. There's also some, I'd say, ongoing discussion about whether fluoride may compete with iodide for transport into the thyroid via the sodium iodide symporter. That 2006 National Research Council report said no. Other researchers have brought forth evidence since that suggests it may. So I would say inconclusive on that from my perspective. There are studies showing associations of high and low fluoride exposure with thyroid disease and changes in thyroid hormone levels. However, the studies that include low exposures are predominantly ecological and cross-sectional. So these are well-conducted studies, in many cases, but there's limitations with the cross-sectional design. And there really is a need for prospective high-quality studies examining associations of low-level fluoride exposure with thyroid outcomes, whether that be thyroid hormone levels or diagnoses of thyroid conditions. Here are two recent studies that are cross-sectional conducted in the MyRic cohort. So this is also the cohort that examined associations of prenatal fluoride exposure with child IQ. They found that higher fluoride exposure among pregnant women was associated with increased thyroid stimulating hormone levels. So elevated thyroid stimulating hormone levels, or TSH, can be a marker of declines in thyroid gland activity. And they also found that higher fluoride exposure in pregnancy was associated with more hypothyroidism diagnoses in these women, but again, cross-sectional study designs. There was a study published about 10 years ago that was an ecological study that found that higher fluoride levels in drinking water were associated with more hypothyroidism diagnoses in England, but again, didn't look at individual measures of exposure assessment. And then a study that I led that was published in 2019 that examined fluoride exposure and thyroid function among adults living in Canada. And we found that higher urinary fluoride levels in this population were associated with higher TSH levels among adults who had moderate to severe iodine deficiency. So that's an example of a study where we bridge, fluoride exposure or an environmental toxicant exposure with looking at status of a nutrient. And of course, iodine is particularly important for thyroid function, which is why we looked at whether that was modifying the association of fluoride exposure with thyroid function, and we found that that was indeed the case based on our findings. So that's what I was talking about at the beginning of this webinar, just trying to highlight the importance of looking at, whether dietary factors may modify associations of fluoride exposure with a health outcome. There's some literature suggesting that calcium intake from the diet may impact how fluoride is metabolized and how that can impact health. For example, also the alkalinity of the foods that we consume may impact that as well. So things to think about. Another health outcome that is, of course, closely linked with fluoride metabolism is kidney function. As I mentioned, fluoride is renally cleared. We excrete it in urine, and it also accumulates more in the kidneys than in any other organ system, which makes sense because our kidneys are, filtering it. Chronic low-level fluoride exposure in animal studies has been linked with quite harmful effects. So an abnormal cell production in the kidneys, reduced enzyme activity, enlargement of kidney cells in the renal tubule, and also swelling and inflammation of certain parts of the kidneys. Studies conducted in China and India also show associations of relatively high fluoride levels with kidney disease and kidney damage. I led a study that was published in 2019, I believe. It was a US-based cross-sectional study where we found associations of relatively low fluoride exposure levels with changes in kidney and liver parameters. Jordana's also led a study looking at fluoride exposure and kidney function in the United States that's currently under review. But we need prospective studies. We need more studies on this topic, ideally prospective ones examining relatively low fluoride exposure levels in relation to kidney function. Lastly, bone health is another important health outcome that relates both to fluoride exposure and metabolism. As I mentioned, most of the fluoride that we ingest accumulates in our bones and other calcium-containing tissues. And fluoride has quite complex effects on bones, and that's another reason why I think it's important for more research to be conducted. Fluoride has been shown to replace the hydroxyl ions in hydroxyapatite to form fluorapatite. Fluorapatite is a more crystallized structure, and this is a similar mechanism by which fluoride can protect against dental caries when applied topically by, converting hydroxyapatite to fluorapatite, which is more crystallized, more resistant to acids. But when it comes to ingesting it and its impact on our bones, fluorapatite, although more crystallized, matures slower. Fluoride can also stimulate osteoblast activity, which can increase bone mineral density. But it may redistribute bone from our cortical bone, which is the hard outer layer of bone, to the trabecular bone, which is a spongier type of bone, and increase risk of fracture. Fluoride has also been shown to delay the initiation and rate of bone mineralization. So this can contribute to reductions in bone elasticity and surface area, and again, ultimately weakening bones. So more research is certainly needed to understand, what the potential effects on bone health at low exposure levels may or may not be. There have been ecological and cross-sectional studies linking low levels of fluoride exposure to an increased risk of bone fracture. So even US-based studies, there have been many conducted, but they're generally ecological. So again, not looking at individual measures of exposure assessment. And most of the studies that have been conducted in this area link fluoride exposure to increased risk of hip fracture or forearm fracture. There was a recent study that was published, I believe, within the last few years. I think it was 2021. I don't see the date on this, but it was conducted among postmenopausal Swedish women, and it had both a prospective and cross-sectional design. So the findings showed an increase in bone mineral density of the lumbar spine and femoral neck cross-sectionally, and an increased risk of major osteoporotic and hip fracture prospectively. And what's interesting about this study is that the water fluoride concentrations were approximately 1 milligram per liter, which is quite close to the recommended level for fluoride in drinking water in the United States and Canada. So we would consider this to be relatively low population relevant exposure level. And I have a grant under review on this topic. I'm pursuing other grants to study this in the US population as well. But certainly, you know, more research is needed, and including in other countries with low exposures or different sources of exposure as well. And the last bone health outcome I want to highlight is bone cancer, osteosarcoma. This, I would say, is probably the most equivocal health outcome in terms of fluoride exposure and bone. There have been associations demonstrated in animals between excess fluoride exposure and bone cancer. There's a biological possibility for fluoride to impact bone health, as I've discussed, given that it accumulates in bone. There have been studies linking fluoride exposure to bone cancer in boys, but the entire literature, when taken into account, most studies do not show an association of fluoride exposure with bone cancer. Most studies are also quite methodologically limited. There was a recent review on this. I've linked it below. I've cited it below, the 2025 study that really highlights all of the limitations in all of the studies. And it is a challenging health outcome to study because it's a rare health outcome. But I think, of course, so important to have a better understanding with well-designed, good quality studies. And maybe even a case control design, given the rare outcome, could help to address the gaps in this literature. So let me just highlight the last thing to highlight here. And I also want to say, if anyone who's attending the webinar now is interested in any of these health outcomes, but you maybe haven't conducted research on fluoride exposure and health thus far, I'm certainly happy to consult and open to collaborations on any of these topics. Lastly, as I mentioned in the beginning, considering co-exposures to fluoride is so important. This is a study by Mullinex, published in 2014, that looked at metal co-exposures in fluoridation chemicals. So this is particularly relevant to people studying participants living in fluoridated communities, because, as you can see, the findings of this study showed that the fluoridation chemicals can contain metals, including arsenic, lead, and quite high levels of aluminum, particularly in sodium fluoride. So hydrofluorosilicic acid and sodium fluoride are chemicals commonly used for community fluoridation. And this analysis found that they contained other metals. So research that examines potential mixtures of fluoride with metal co-exposures, or other co-exposures, as I mentioned, depending on the source of fluoride. If fluoride is coming from the diet, then the co-exposures may be different. But we welcome any submissions related to this area. Thank you very much. That concludes the webinar presentation portion for today. And we welcome any questions.
Yeah, and we do have questions that we've been writing some answers to in the chat that I think we could read and answer. So
Sure.
someone asked about IQ as an outcome measure. Is it a legitimate outcome measure? From my knowledge, IQ has been a standard method for measuring cognitive abilities, and is used in regulatory agencies like the EPA when assessing chemicals like lead and other neurotoxicants. It's a very highly reliable predictor of later life outcomes like education, occupation, physical health. So it is, I think, a very important measure that has been used for a long time and is still being used. I don't know if Ashley, if you have other insight into that question.
Yeah, I agree with what you said. Psychologists do assess IQ in children, at the ages of 3 and 4 as well. I think that's been a question. But yes, I agree with that.
Mm-hmm.
Okay. I see a question from Hardy Lineback. To all other than Till et al. In Environment International 2020, are there any studies that show increased fluoride exposure affects developing brains after birth? Okay. This is relevant to the current controversy on FDA wanting to remove fluoride supplements from the US market. Yeah, that's a great question. So, of course, just to fill everyone in, Till 2020 looked at fluoride exposure among infants in the first 6 months of life from infant formula and found it was about a 9-point decrease in IQ among the infants that were fed formula reconstituted with the tap water for each 0.5 milligram per liter increase in fluoride concentration in the water. So that points to an association from infant exposures. In terms of childhood exposures, as I'm just reflecting on the literature, there's inconsistencies there from my understanding. I'm thinking of there's studies showing cross-sectional associations with increased diagnoses and symptoms of ADHD. There's other studies I think that don't show associations with IQ from childhood exposure.
Yeah, I think it's hard to separate. A lot of studies don't adjust for the maternal exposure or for the gestational exposure.
Okay.
Studies that do measure children's exposure and children's IQ. Yeah, so I don't know if it's really hard to tease apart that prenatal exposure. I know the Till study did adjust. They did adjust for the maternal urinary fluoride in theirs, but I haven't seen that. I don't think that's very common in the studies that we've looked at, so it is difficult to.
Yeah. I think there's enough consistency in the literature from prenatal exposure to suggest that that is a, sensitive, susceptible window for potential developmental neurotoxicity of fluoride. Same with, the infant study was quite compelling. Yeah.
So there's also a question about what does it mean for an individual or a population to have a 1 to 5 IQ point drop. So also from my understanding, a difference in IQ of a few points for an individual may not make much difference in their life. I personally would not want to lose a couple of IQ points because I want all of the ones I have. At a population level, however, these few points do have a huge impact with a lot of implications. So very subtle shifts in IQ, what we know from other neurotoxicants like lead, mercury, organophosphate pesticides, is it really can shift that distribution, and that has an impact on kind of the people with the highest IQ and the lowest IQ. There is a great graphic of that. I don't have that available though.
Okay. And, I see, hold on, there's more questions coming in.
There was some question regarding Mardra's cohort. They were asking like whether we have IQ test or other cognitive test already been obtained. So yes, we conducted. The project that I just shared, ASQ score is already like measured by Mardra's and also SRS, IQ related. They have 5 domains, so yes, they have covered. And also they asked about, has the Madras cohort, which is most Hispanic resident of Los Angeles, experienced difficulties with participant retention given the current problems for immigrant population? So yeah, I think specific retention rates, they are not publicly detailed, but I think or any cohort that involved vulnerable population, they required enhanced engagement strategies such as bilingual support and culturally sensitive communication. So I think Madras is good at this. They have very well-established team, and yeah.
Yeah, exactly. And Madras does not have IQ tests, so there won't be a Madras fluoride and IQ
Oh,
study anytime soon, just to answer that part of
yeah.
it.
Okay.
That's a common question that we get. We have all the other measures that Jordana mentioned, or they do, rather.
There was a question about genes, like if there is any, what genetic factor are
Oh.
suspected to lead to adverse effect of ingesting like fluoride by pregnant women or young children. So I saw some studies which looked at like genetic variants, specifically those with the fluoride metabolism. So some people have different gene set that involve detoxification pathways. They can influence, like for different people, different level of fluoride intoxication.
Yes, and there's also a really nice study by Kelly Brunt's group that I'm just going to link in the chat, that they looked at an epigenome-wide association of fluoride exposure and DNA methylation in US children, and found that fluoride was associated with some epigenetic changes. So I'm just going to link that.
Another important question that I got was, how should these potential risks be weighed against dental public health? And I think that's kind of the question. Altogether, our research is focused on the risks. We did not consider benefits in our work. I know that the FDA meeting on supplements and others are trying to understand more of that balance. And I think the balance based on scientific evidence is very important. A comprehensive public health risk-benefit assessment of fluoride exposure with the risks and the benefits for dental health is definitely needed to inform, I guess, recommendations or public health moving forward.
Okay, we have another question. What about individual hypersensitivities to environmental chemical exposures? Are there any recent studies about such sensitivities to fluoride exposure? That's a good question. I'm not aware of, well, I mean, hypersensitivities can be interpreted in different ways. I'm not aware of any studies that are specifically looking at that. Kyla, are you?
I don't know if that includes the genetic mutations. That's what I would think of, which there are.
Okay.
See, there's a question about how long supplements were recommended for pregnant women. This was a common practice in the US and Europe. Yeah, I don't know that. I don't know. I know that was something that was recommended. And potentially, since people are getting more fluoride exposure from multiple sources, even in Europe, if they're drinking lots of tea or then they're already getting that systemic exposure. But I don't know when recommendations for that ended or when they started.
Yeah, and there was a study conducted at Johns Hopkins that was administering fluoride supplements to pregnant women. It was a clinical trial, and my understanding is that was halted after some of the studies published on fluoride exposure and IQ came out.
Okay.
So, yeah. I'm not sure how the recommendations have shifted in the last decade.
Yeah. There's a question about how similar is this research to lead, I guess for the neurotoxics. I know what we're seeing for fluoride exposure in children's IQ has a lot of similarities with, especially the standardized mean differences that's very similar to early lead exposure research. It's the first step in identifying a hazard is looking at very high exposures compared to low exposures, which is what the earliest studies in fluoride and I think in lead did look at, which identifies kind of that association. And then as more research comes out at lower exposure levels, we kind of fill in that information more and more. There is, of course, a major difference between lead and fluoride, with fluoride having dental health protection, whereas lead, there's no benefit for that health-wise. So I think that's something that we're still, for very low levels, that's something that we still need more information on to understand. But I would say that there are a lot of similarities. Let's see, in the chat, someone asked about effect fluoride might have in adults. Our NTP monograph did look at cognitive effects in adults. There are very, very few studies that looked at this. And they had different, they used different outcomes that weren't really comparable. And I think they had conflicting findings. So I think that is an important question, and something that future research should also consider.
All right, I'm just seeing if we missed any other ones. Does fluoride accumulate in the brain? Yes, there is evidence that fluoride can cross the placenta and accumulate in regions of the developing brain. All right. Anything else? I've left my email. Well, my email is ashleymalin@ufl.edu, and I think it's linked on the materials for this webinar. So if anyone has questions, you're welcome to reach out after this. I believe this webinar is coming to a close. Thank you everyone for being here. And again, to our speakers, Dr. Kyla Taylor and Dr. Jordana Khan, for presenting on this important work. And we appreciate everyone who attended today. And thank you. Thanks for having
and for
us.
your great questions today too.
Yes.
Thank you so much.
Thank you.